Before I go ahead, I would like to thank ISMB and Function for this opportunity. Precision medicine is one of the most trending subjects in life sciences today. And this initiative has been taken by the former President Barack Obama in 2015. And that's what my lab is trying to learn and practice at Rutgers. Talking about precision medicine is mainly about investigating one patient's variable kind of data, which includes, for example, clinical, multiomics, and lifestyle. Whenever a patient visits a hospital, different kind of diagnosis is performed, which includes conducting different kind of lab tests, and if required, provided different medications and procedures. However, the question remains there is if one medication which is good for one patient will be good for all of the patients with similar symptoms. To investigate this question further, we look into the different kinds of genomic data, for example, expression and variant. And we also are interested in looking into the details encapsulated within metabolomic information or data like carbohydrates, amino acids, and lipids, etc. This whole information evolves throughout the different layers of this information evolves different levels of the layers of data. And another question is how this data is generated and efficiently can be integrated and processed. So this data is generated, as we all, most of all, at least now, that different institutions or different units within one institutions or collaborating with multiple institutions are working and generating variable kinds of data, with the challenge around that these data is of different kinds, different structures, and heterogeneity. This is one of the challenge my lab is addressing at Rutgers, that we are focused on integrating different kinds of data together and practice precision medicine. One of the kind of those projects is dealing or studying heart failure. Heart failure is one of the leading causes of death in United States and also in around the world. So what we are doing here is we are focused primarily on gene expression and variant data only. What we did during this study is we started consenting patients visiting the hospital in outpatient pavilion. And we started collecting blood samples randomly. Then the challenge was, okay, which samples are related to the CVD patients? For that, we developed a tool which is called MAVClick. It's open and freely available, open access and freely available tool. This tool actually helped us in connecting our system to the Epic health system in the clinical environment and link our sample data with the Epic and investigate which patients from our overall cohort belongs to the CVD diseases. Second step was about definitely sequencing those patients. We generated whole genome sequence data and RNA-seq data. And then we started investigating or performing downstream analysis, which started and ended with the gene disease data annotation, interpretation, expression, and variant analysis. Please keep remember throughout my talk, I'm talking about same patients with different kinds of data. So for that, when we started gene disease data annotation, we started building a unique database, because for referencing about the non-human gene disease information, we needed that. So we studied several databases available around. This database, which we have developed now, is a combined form of variable databases around, and this is also freely accessible and published published in an article and freely accessible through smartphone apps. So the first part of our study was about gene expression analysis. With that, we developed, my lab is a 2 years old lab in Rutgers, so we are not very old. So when we started, we never had pipelines. So we started developing our pipeline for gene expression data analysis. And the pipeline is not unique. Most of the people around the world are practicing the similar kind of pipelines. However, one of the unique part of our pipeline was introduction to the GWIS. GWIS is our own produced tool in the lab, which supports, without much bioinformatic knowledge, a kind of a gene disease data annotation, enrichment, and expression analysis by linking clinical transcriptomic variant and gene disease annotation data together at one platform. One example you can see is this big heat map. So here in this heat map on the right side, you can see all the genes we have found associated with the heart failure disease and some other CVDs. On the left side, it also identifies all the diseases from the clinical data linked to these genes, and for those patients which are on the right, on the bottom of the heat map. You can clearly see the difference between the patterns in this figure. On the right side, we have all the control samples, or on the middle to the right, to the left is or all the patients diagnosed with different kind of CVD, especially heart failure. So time is short, I'll go through quickly. So we perform some gender-based classification of the data. to identify the significant genes. We performed race-based classification of the data among the black, white, and all other races from the cohort we had. Then we also performed some traditional downstream analysis, which involves differential gene expression analysis, enrichment, pathway analysis. And this all information is available in this article, I'm sorry, which have been already published and available. Second part of the study that we were interested in looking into those, especially those genes which were having regular, significantly high or significantly low expression among the CVD or especially heart failure patients. So we took those samples again from the same patients and then again did the sequencing, which I have already mentioned for whole genome. Then again, we have run into the issue that we never had a pipeline to process and discover the variants. So we developed another new pipeline. That pipeline was again integrated to our previous pipeline, and we start discovering the variants. So this pipeline is also published and freely available and accessible. Then we started computing, first of all, the mutations per percentage for heart failure patients and other CVD. And also we started computing mutations among those genes we were really interested in. We performed different kind of variant analysis. One of them was circus plots, and here you can see we have displayed this circ plus is displaying all the samples having mutation among all those genes which were reported by our GVIS tool associated to be the heart failure patients, and these are with the other CVD, and these are all which are reported to be having significant expressions classified by gender and classified by their race. Then we also perform functional mutation and non-functional mutation analysis. And we used an interesting tool, which is already published, which is called Mutation Mapper. We tested it. We were interested in doing our own downstream analysis. However, we were also interested in using some gold standard tools around so we can verify our results are correct or not. So we're not just running alone in the jungle. So this is what we did for CVD as well. So I have only 10 minutes, so I want to conclude on time. So the paper is already in PubMed. I will request you to have a look at it if you're interested. And one other paper, which is based on integrated gene variant analysis with expression data, is in review and hope to have it soon outlined. So summarizing it, I conclude with primarily, I have studied everything summarized already, but I would say we were able to identify altered expression of 4HF and other CVD genes, which are these four. So going forward, we are focused on implementing AI. Recently, we have published a very interesting article with one of the top journals in bioinformatics, and this is about implementing AI only on gene expression and variant data. So we are practicing right now random forest and SVM on the same data of the patients, and we are leading our step next to do some predictions using the same data, because we are very fortunate to have a data of these patients of different kinds, so it will leverage us in different ways. This is my handsome lab. So I have some students at Rutgers, a brilliant institution in the United States, and the brilliant students from that institution, and some collaborators from UConn and industry. And if you have any questions, you're welcome.
Thank you. We do have time for one question, maybe.
Can you repeat the question before answering so that the virtual audience can hear?
Hi.
So thanks. That was interesting. I always wanted to know what you do exactly. We're just. So my question is, there has been a lot of study on variant burden, including some work from Iraq. I'm kind of curious, have you compared your performance?
I'm sorry, I never understood the last part of your question.
Have you compared the things that you did?
Yes. Okay, that's what I was just mentioning. We did comparison, and not with your lab, but some other work which is more close to our because today here I'm only talking about CVD, so we were more focused on only those literature which are publishing gene expression and variant analysis on CVD. Genes. So definitely we did, and we reported actually some articles on that as well. And for that, one of the article is also in review and coming. So that's it. Yeah, and also we have used some tools which are publicly available. Like for example, if I'm doing gene expression analysis using my own tool, and at the same time we are also using some gold standards, so we are comparing the overlapping between the results, and that's what we're also doing for AI as well. Like for example, if we are implying random forest, just imagine random forest is not a part of RSEM or GTech. So if a result or mutation based results or expression based results are coming from those tools, how much overlapping is there with the AI methods? So, and there is significant, some genes are overlapping highly. So